As far as the organization of genetic material in bacteria is concerned, they typically have a single circular chromosome. A single circular chromosome is present inside the cell, and this chromosome consists of a single circular molecule of DNA. This DNA is associated with some proteins. The bacterial chromosome is very long because it consists of a single molecule of DNA, and it may be up to 1 millimeter long, which is several times long than the length of the cell. So this DNA molecule is looped and folded, and at some points it is attached with the plasma membrane of the cell. For example, here you can see in this cell, bacterial cell, this bacterial chromosome is present. You can see the chromosome is folded and looped at various points, and it is also linked or attached with the plasma membrane at different points. So this is the chromosome of a bacterial cell. So let us talk how this single molecule of DNA is organized into a chromosome or into a chromatin body, which is called nucleoid. So specific proteins interact with the bacterial DNA to form a highly condensed nucleoprotein complex, which is called nucleoid. So different proteins, they interact with the DNA molecule, and they form this highly condensed structure, which is called nucleoid. So a nucleoid is present in the cell of bacterium. Bacterial chromatin can be released, this chromatin or nucleoid that can be released from the cell when we gently lyse the cell. For example, when the cell is gently lysed or broken down, this nucleoid or chromatin can be released from the cell, and this nucleoid can be visualized under the electron microscope. And if we see it under the electron microscope, it reveals that it consists of multiple loops which emerge from a central region of the chromatin. So there is a central region in this chromatin, and many loops, they emerge from this central region. For example, some of the loops are supercoiled while some are relaxed. So in this chromatin, some loops are highly coiled while some are comparatively relaxed. Relaxed loops, how they are formed? Relaxed loops are formed as a result of a nick or a break introduced into a supercoiled loop by a cellular DNase, because in the bacterial cells, DNase is an enzyme that breaks down DNA molecule. So these DNases are present in the cell, and sometimes these supercoiled DNA, they are nicked by these DNases. So if a supercoiled loop is nicked, it becomes relaxed loop. For example, here you can see this is the electron micrograph of chromatin, and you can see in this chromatin there is a central portion, and from this central portion several loops are emerging. In this representation, you can see this is the central core of proteins, and several highly condensed or highly coiled loops are emerged. Here, this loop, you can see this loop, it is relaxed loop. So sometime this was initially supercoiled, and sometimes DNase nicked this loop and it became relaxed. How these supercoiled loops are formed? Because the DNA molecule is actually a linear molecule in the bacteria. So look at this. This is the DNA molecule which is linear, and then it becomes circular because a circular DNA molecule is present in the bacteria, and then this circular DNA supercoils, and if the supercoiling continues, it becomes highly condensed. So in this way the DNA molecules are highly condensed in the chromatin.